Okay. Hi, everyone. Welcome to this is the fifth seminar of the MaLaMoBi series, the Mathematical Laws of Morphology and Biomechanics. It is my very great pleasure to introduce Professor Angela Horner for this week's seminar. She is an associate professor in the Department of Biology at California State University in San Bernardino. Her research group focuses on mechanisms that shape locomotor strategies in animals and how proximate and long-term perturbations affect performance. So she's done a bunch of work looking at how changes in substrate in the environment, but also things like aging and disease impact locomotion and in vivo muscle and tendon function in a variety of vertebrates, but with a particular focus on some small mammals and crouched postures. So without further ado, I'll hand over the screen to Angela so that she can tell us about when the product is not the sum of its parts and missing links in locomotor biology.
Thanks. Try to do this as seamlessly as possible. Okay. Thank you, Kathleen. I remember when Kathleen was just a wee PhD student. She wasn't so wee. She was regular size. But she came in and did a biomechanics talk for my class, and that was a great insight into some of the work she's doing, and it's just so cool to see her at Ball State with her own lab. It feels like just yesterday. Okay, so I am at Cal State University San Bernardino, and I want to talk a little bit today about when engineering goes wrong or when perturbations in the system create havoc with the predicted or modeled outcome. So in the case of this robot who's encountered a perturbation in the substrate and did not compute, that's going to result in a failure. That robot is going to fall to the ground. In this famous FIFA glitch, this is probably a few lines of code that could be adjusted. But again, when the modeling or predicted outcome doesn't work out quite right, we see something like this. In biology, for an organism, such failures have much more dire consequences. So I want to briefly sketch out kind of my take on animal locomotion and my approach. And I do admit I'm a very gestalty kind of person. That is, I am constantly living in big picture, and it takes collaborators and good advising to bring me down to kind of the mechanisms and the small nuts and bolts. But together, I kind of put together this version of what my take is on animal locomotion. So organisms, of course, have to exert force on their external environments. And so we have Newton's second law and related functions, force equals mass times acceleration. There are particulars in there that cannot be altered. We can't change gravitational acceleration. None of that is under biological control. But over evolutionary time, tissues can be adapted to some extent, and we can work on other aspects of these equations. But here's the catch. Tissue adaptation is still limited by biology, so we can engineer a fantastic system with none of these sort of limitations. But in biological systems, we are limited to developmental constraints, phylogenetic inherited characteristics, parturition or birth. We can't have bigger heads because we would wreck mom's pelvis, that sort of thing. So in biology, we have these amazing systems where we have analogous functions that is something to move for locomotion, and it is derived from homologous parts. And then we have to also, when we're dealing with these big pictures or big projects in animal locomotion, we have to also consider size and scaling. And certainly in my training, we spent a long time thinking about what kind of physiological traits, how they scale over body size, particularly in mammals. And so we have all kinds of measures of performance we look at, from what we call a mouse to elephant curve. It could be maximal running speed, it could be VO2 max, any sort of thing like that. However, we know that there are some things that have to change with that scale. For instance, we have an enormous elephant with pillar-like columnar limbs that have to be oriented directly under its body to support its great weight. Whereas a water strider, in order to maintain its ability to move over water and take advantage of surface tension, actually has to have its limbs spread very far apart. We don't need math to intuit that those two different postures wouldn't work in the other's environment, but we do have math to back that up. One of the problems that started to puzzle me more during my graduate degree was paying attention to the little guys. So it turns out when we pull out smaller mammals, they tend to have a different scaling relationship in and of themselves. So we lose a lot of that if we're looking at big scales. If we look at a mouse to elephant curve, we're losing a lot of the nuance in these small mammals. So I have pretty much taken upon myself the last 10 years or so to look at these homely microfauna. And they are certainly no engineer's great dream. There are all kinds of things that are wrong about them. Because of their crouched posture, they have decreased limb mechanical advantage. Because of their shorter limbs, they have to take many more steps to go the same unit distance, and that costs more energy. And because of their small size, they don't have big enough or strong enough tendons to store meaningful amounts of elastic strain energy. So why be the way that they are? There are all sorts of different reasons for that, and a lot of it has to do with being able to hide from predators, among other things. But it also makes them exceedingly difficult to work with, especially if you compare these homely microfaunal animals to something like a dog or a horse. So the way we measure performance at an organismal level is we can look at force plate recordings, look at how their body is interacting with the ground via force plates, basically like a big fancy bathroom scale that measures in 3 different directions. We can look at high-speed video and capture things like gait, posture, and kinematics, as well as looking at whole animal metabolic rates. Measuring tissue level performance is even more difficult with these smaller animals. So we can use things like indwelling electrodes to measure muscle activation. We can put something like sonomicrometry crystals in muscle fascicles to record length change in a muscle over time and record the force transmitted through a tendon during locomotion. All of these things have to be implanted into an animal and usually tied to a cable, not always. And again, this is really exceedingly difficult because it tends to interact with their locomotion. However, this whole field of comparative biomechanics has really benefited a lot from people who are able to creatively think in engineering terms, being able to apply some engineering principle to an existing biological phenomenon. In this case, we have a sling jaw wrasse, which is able to protrude its jaws exceedingly far from its own body. Folks more creative than me were able to link this to something called a 4-bar linkage system, which is a pretty common mechanical system, and be able to model jaw protrusion using this 4-bar linkage system. And I would say in the last 10 to 15 years, animal modeling and biomimetics has really exploded as a field. And we've taken some really interesting animals and been able to model them as robotic organisms. To me, one of the most interesting aspects of this is that it also exposes some of our gaps in knowledge. So we know that muscle is a motor. It actuates a system. It can have in parallel or in series elasticity. We have the activation system usually well known. We can predict with great accuracy what the outcome, what the performance should be. But there are still some gaps. For instance, here's a galago or bush baby hopping towards you. Remarkably, these animals can also jump up to and over 2 meters in height. So they are such exceedingly great performers. They have been targeted by biomimetics researchers to try, that's my dog, I'm sorry, to try to model that same kind of amazing jump performance. So jumping robots is actually very difficult to do. And one of the recent takes on building a better vertical jumping robot system decided to actually, let's see if I can pull up my laser pointer, add this important component. So we have the motor actuator and then an in-series elastic spring. And this is incredibly important. Muscle is only capable of producing so much power, but if we add an in-series elastic element, we can store and release that power, or store and release that energy to produce more power. So using a jumping robot, they were able, in this red line here, to capture 78% of the animal's performance. Now, 78% as a biologist sounds okay, but to the robotics industry, this is great. This is what previously performing jumping robots were able to do, not having that in-series elastic element. But for me, I'm constantly intrigued by these gaps, these differences in what the model and what the biological outcome have actually shown us. And I'd like to go through two particular problems which I've been working on for almost an embarrassing number of years, but anyway, some number of years. And the first one has to do with the provenance of age-related loss in muscle force production. We call this phenomenon senile sarcopenia. The second case study will be an examination of something more comparative biology rooted, that is the efficiency of locomotion in an inefficient posture, that is during tunnel locomotion. So first of all, I'll just try to take us through this very briefly, but muscle tissue, we have to kind of understand the components of how muscle produces force to understand why we should expect to know how much force senile muscle should produce. So muscle tissue has to consume ATP to produce force. The amount of force it can produce is a direct product of how many cross-bridge cycling we can produce between actin and myosin. And this muscle force is also related to its length. So if we pull a muscle, or at the sarcomere level, this scales up as well as back down, at different lengths, a muscle sarcomere or a whole muscle can produce different amounts of force. The amount of force that is produced is a direct proportion or directly related to the amount of actin and myosin overlap within the sarcomere. So we call these parts of the length tension curve the ascending limb, the plateau, and the descending limb. And we can pretty much nearly correlate that to the amount of actin and myosin overlap. If we pull our muscle too far, we start to see less actin and myosin overlap, and then we get into a territory of perhaps doing damage to the muscle. So that's the descending limb. We generally don't want to operate on that part. This ideal region we call the plateau. So this is the optimum length for a muscle to produce force, and it has the optimal overlap of actin and myosin. In invertebrate muscle, this is highly conserved and tends to correspond to a sarcomere length of about 2.2 microns. There's very little deviation from this, but there is some, and that's also interesting. Not nearly as cool as inverts, where they have many, many orders of magnitude in length differences. The muscle force, because of this relationship to cross-bridge cycling and actin-myosin overlap, is proportional to its size or its cross-sectional area. So if we have a simple parallel fibered muscle, we will expect this very thick, wide muscle to produce more force than, for example, this very long muscle with a smaller cross-sectional area. So if we want to produce more force and we aren't worried about any other constraint, such as the energetics of having a big muscle, etc., we would want to build a wider, thicker muscle. There are all kinds of biological constraints in the system for that, right? We can't just keep on building wider muscle. That would be extremely inefficient in terms of hauling our mass around. So we frequently encounter pennate muscle. That is, muscles with fibers at an angle relative to the line of action of the whole muscle tendon unit. These tend to have a shorter functional distance, but can produce higher force because we can pack many more fibers in to increase our cross-sectional area, or our physiological cross-sectional area, now that we're taking into account that pennation angle. Okay, so now we have all the elements pretty much we need to predict how much force a muscle can produce. But then we have the case of senile sarcopenia. So as we age, all mammals, all vertebrates, for the most part, our muscles are going to decline in performance. This is regardless of your physical status, regardless of how well you've trained your whole life, you can slow the process down somewhat if you're an elite athlete, but you cannot prevent it. So muscles have been shown to reduce in mass by quite a bit. This is not a surprise. This is going to be responsible for a lot of the decline in muscle force, but not all of it. We also see reduction in fine motor control, the sad declining ability to repair muscles, so you can still continue to do strength exercise and workout, but you'll not reap the benefits quite as well in your old age. Related to that, we see decrease in pennation angle, decrease in joint range of motion. This is just kind of a laundry list of declining locomotor ability of older mammals. But then there's a muscle weakness that is not directly proportional to this reduction in mass. In fact, in mouse studies and in human studies, we can see that the reduction in muscle performance actually precedes this loss in muscle mass. So this is a huge confounding problem. There are a great many of people in the biomedical field that are interested in this because they're interested in human health. I myself am more interested in how come this is the way that this is. As an example, we see in these data from young and old rats, for example, a force frequency curve where we can see muscle force at these various frequencies. And not surprisingly, young muscle is able to produce more force at a given frequency. When this is controlled for body weight, the aged rats tend to be kind of big and tubby and not very lean. This is not so surprising. But if we take it as a measure of muscle quality, that is force produced per muscle mass, we see that there's still this pretty large difference. So again, what this alludes to is that there is some other factor that is producing lower force in aged muscle that is not simply mass alone or cross-sectional area alone. So one of the things that started to intrigue me is this idea of a muscle's operating length. So we know that muscles can only generate force at particular lengths. If they're too short, they don't have enough actin-myosin overlap, so they can't produce as much force. If they're too long, we actually start to see some damage occur. So animals tend to stay away from these longer lengths, except in some amazing examples. So a study that Manny and Tom Roberts did years ago showed that some frogs are actually able to start off their muscles at a very long length. And the reason they're able to do this is because the passive tension of the whole MTU is quite elastic, it's quite slack. So without doing damage to the muscle, they're able to start at very long lengths. And as the muscle is contracting and shortening, it ends up at a very optimal part of the force length curve. So here we have an example of how organisms can modulate operating lengths to optimize force, but perhaps this might be done in a negative sense too. So if we have, for example, young and healthy rats and versus aged rats, we know that the aged rats should be producing less force, but where will they be operating at on that length tension curve? Could be that they're using similar operating lengths, and this has nothing at all to do with the observed differences in vivo force production. Or perhaps they operate at relatively longer lengths. So they might be doing this not because they want to, that's kind of a terrible way to talk about things in biology, but rather because there's simply an absolute distance that the muscle has to work at to make a joint work, or to produce a good enough movement at the joint. And so at these long lengths, they're of course in danger of doing some damage. So that might be limiting force production in different ways. Or they could operate at relatively shorter lengths, thereby staying away from these potentially longer lengths that could cause damage. So these are the hypotheses going into the system. We used fluoromicrometry to measure this in aged and young rats. I'm not going to go into too much detail about this here, but recall that I did say small mammals are notoriously hard to work with in terms of instrumentation. Any kind of little device you put on them their locomotion, their behavior tends to change radically. But fluoromicrometry, we're able to put these little tantalum beads that are radio opaque and put them right in the muscle so we can measure muscle strain in vivo and can compare that to in situ properties as well. So here we see a video of a young rat moving nicely across this trackway. You can see the constellation of tantalum beads in its medial gastroc there. And then compare that to an older rat, which is having such a hard time it might crash the computer. So this is pretty typical. There's nothing, I just want to point out that this is absolutely corroborated by many other studies. We see differences in posture, we see differences in voluntary speeds, much shorter steps, all sorts of things going wrong. This is a very old rat, by the way. 32 months is very close to a rat's natural lifespan in the lab. So we can look at those beads in vivo and see what's happening to the muscle during locomotion. So this gray box represents stance. That's when the muscle is able to produce force, exert force on its external environment. And we see this very typical pattern where the muscle is shortening at the beginning of stance and then no length change for the duration after that. We can compare these operating lengths to in vivo stimulated muscle where we maximally stimulate the muscle to produce force at different lengths. We can map those lengths onto a force length curve. And we can compare what this little bit at the beginning, that's the passive tension of the MTU. So passive tension I didn't describe too much here in detail, but it's a combination of like extracellular matrix, parallel elastic elements in the muscle as well as the tendon. And all of this is contributing to what we call passive tension. And if we pull the muscle very far, we'll see that passive tension rise. So once we generate a force length curve for individual animals, we can map those in vivo strain lengths onto our force length curve to see where on that force length curve an animal is operating in vivo. Long story short, if I take out an individual that is problematic, which I'll talk about in the next slide, we see a general pattern that fits kind of what we predicted. So the young rats are tending to operate at longer lengths. In this particular case, the y-axis is not important. We're just mapping lengths onto this operating length, onto this length tension curve. So what we see is that indeed the young rats tend to stay pretty close to optimal length near that plateau, and aged rats, at least these aged rats, tend to stay at shorter lengths. Now, I know that Natalie Holt is in the room, and what she can attest to is that we have these just so stories we have for aged muscle studies, and then there is some variation in that. So there's exceeding amounts of variation in methodology, there's exceeding variation in the types of muscles that are used. You can get different results if you're, say, looking at an extensor versus a flexor. And then there's also simple, frustrating individual variation. So in this particular study, we had an aged individual with very passive tissues, or very elastic passive tissues. The nice thing about that, in a way, is that the operating length of that particular individual matched up really nicely with its passive tension. But this is a frustrating field to work in because in the aged muscle biology world, there are so many factors that bring variation to the system that are not always well accounted for. And then even if you do account for all those variations, it still might be an individual variation. So we can sort of say that aged rats tend to operate at relatively shorter lengths in vivo, and sort of say that passive curves in aged rat suggest stiffer MTUs, but we have our exception. But we absolutely can say from these data that operating length can be predicted by passive stiffness, and that is pretty cool and has not necessarily anything to do with the original problem, which is where does muscle weakness come from if it's not simple loss of muscle mass? But what is intriguing about this is that the role of passive stiffness in dynamically interacting with muscle operating length is more important perhaps than we previously have considered, and certainly could be contributing to some general declines in locomotor activity of aged individuals. I'm going to try to speed this up. In the case study 2, this has to do with an old Just So story from, I believe it was Journal of Mammalogy. And this used to be one of my favorite things to do. And I don't want to sound like an old person, but honestly, if you can just go to a library and randomly browse some journals from, like, I don't know, 1960s, there are some really cool things in there. People used to be allowed to publish studies of, say, I was walking in the garden and observed a mole searching for a grub. And then that's a paper. Like, they describe it, scientifically. But there are some amazing things in there. So one of these Just So stories is that weasels and weasel relatives are weasel shaped. They're tubular, they're snake dogs. They're weasel shaped to be able to fit into tunnels. And they can fit into tunnels and then eat the things that are in there. And that's why they are the way that they are. And one of the things that made me question that is not being a mammologist myself, but taking a mammalogy course and realizing that if you're a mammal and you're under 5 kilos in size, you're going to spend some time underground. Almost all mammals do, and very few mammals are weasel shaped. And it's not a particularly good shape to be if we are strictly thinking from a biomechanical engineering perspective. That is, there's higher conductance, loss of heat because of the surface volume to area ratio. When you're long and tubular, there's a greater distance from your center of mass to the limb girdles, thereby increasing the moment about those girdles. And the relatively short limbs mean, again, we have to cycle our limbs even more to move a given distance, and that's terribly inefficient. So let's try to break that down quantitatively. Why is this posture inefficient? And what I'm going to show you here is a comparison of a ferret when it's moving around above ground and a ferret when it's moving in the tunnel. Now we see that, okay, being long is probably good in some ways, but one of the things that's not going to be good is that stretching the functional trunk length, increasing the distance to the center of mass. Additionally, we also have to crouch our limbs even more. The more our limbs are crouched, the increasing distance of our joint to the ground reaction moment is going to mean that more muscle force is going to have to be used to counteract that. So that's not good. And again, here we have a rough estimate of the center of mass in this position and the center of mass in this position. And by the way, there are different ways to calculate the center of mass, but one of the classic ways, which I did in this particular case, is to take ferret bodies and pose them and freeze them, and then you measure center of mass in the old way, which is to balance on a balance board. We can also calculate the center of mass mathematically using force plates and inverse dynamics, but this is more fun. So as we increase the distance of the center of mass between the girdles, we're increasing the moments about each of those girdles, the hip and the shoulder. And a lot of that muscle, or a lot of that has to be counteracted by epaxial and hypaxial musculature. So again, all of these suggest that this posture in particular is going to be metabolically more costly. There should be more muscle force that's required to counteract these increasing moments. Therefore, it should be metabolically costly to be in this particular position. So just a brief overview comparing ferret performance in tunnels and out of tunnels. They do remarkably well. Again, this is all voluntary locomotor activity. It's very difficult to get a ferret to do anything unless it's voluntary. So we see that in terms of performance, there's not much of a difference in speed. And it turns out there wasn't much of a difference in any other of our classic organismic level performance variables. So there was no difference in velocity. Again, these are voluntary preferred speeds. No difference in the type of gait used, duty factor, step length, stride frequency. None of those things were different, but there was a huge difference in the posture. So they were able to maintain performance for all intents and purposes, but certainly this should be costly. So I measured the metabolic cost of these activities. This particular video might not work. Apologies, but basically what you'd see is a ferret licking peanut butter at one end of a respirometry chamber, and I promise that it's able to gather more than just the backside of the ferret. So all of the gases are mixed in the chamber, and we pull at a high enough rate to sample all of that, and we match the treadmill speeds to the preferred speeds the ferrets were doing in overground studies. And this was published in the Journal of Obvious Biology, but it turns out, just kidding, it was experimental biology. But it turns out that it does indeed cost more metabolic energy to move in a tunnel than overground. Again, not surprising. That's exactly what I expected. However, what was surprising is that if we compare these values to one of the mass-based equations, predictive equations generated by Taylor et al. In their phenomenal study in the 80s, where they measured all the animals practically and measured the metabolic rate of almost all the animals moving on a treadmill, they generated these mass-based metabolic equations, and this is the line that represents what is predicted for ferrets for the cost of transport. Now, note, cost of transport is the amount of energy per unit mass per unit distance. And so this is a way we can easily compare many different sized animals in different conditions. And this is what is predicted, but for most of the ferrets' preferred speeds, they're falling well below that line of prediction. And as is my desire, I tend to focus again on animals that are a little bit small. So this is a sub 10 kilogram scaling relationship, which is different than a mouse to elephant kind of scaling relationship. I combed the literature and found all the animals I could, particularly mustelines, and mapped them onto this curve. And what I found was that for our guys, Empatorius furrows, the domestic ferret, even in the tunnel, they were falling well below that fit. For example, just to give you a little bit of frame of reference, Glaucomes is the flying squirrel. Turns out they are not so efficient when they're terrestrially moving. That's to be expected. Something called a daegu, which I also used in the study but didn't show those data here, fits pretty well along the line as predicted. But ferrets are surprisingly efficient. Now, why is this surprising? Their postural differences do not suggest that that would follow. So while I was at Brown, I got to overlap with Sabina Moritz, who had happened to have done a PhD on a related problem. So she looked at the epaxial musculature in ferrets, and in particular at the multifidus and the sacrospinalis. These are two muscles located in the kind of lumbar region of the back of ferrets, and they're supposed to be really important in helping to maintain that beam-like posture as the animal is moving through a tunnel. So what she found is that these particular muscle groups had a higher proportion of oxidative fibers than they did fast or glycolytic fibers. This meant that they should be more efficient, and we would also expect muscles with a higher proportion of oxidative fibers to be active tonically kind of over an entire stride cycle. So think of your postural muscles, like our own epaxial and hypaxial muscles right now. If you're sitting up straight, everyone's sitting up straight now, all of those muscles should have a low level of tonic activity during seated posture. So in order to investigate that further, we used EMGs to target the multifidus and sacrospinalis. Normally, if you do an EMG study, many of you in this room probably are questioning this right now, the best way to verify the position of those electrodes is post-mortem. You have to do a dissection and make sure your electrode are in the muscle you thought they were. In the case of the ferret and these multifidi and sacrospinalis muscles, we had some nice bony landmarks that we knew we could hit. So if we hit transverse process, we knew we were in sacrospinalis. If we could line it up against the spinous process, we knew we were in multifidus. So we would implant these electrodes into the ferrets. We would wait hours for them to recover from anesthesia, hours and hours and hours. And eventually they would move. And I want to help us recall what was predicted. So again, multifidi and sacrospinalis muscles have higher proportion of oxidative fibers, therefore they should have low-level tonic activity, in particular in this posture. What we found is that these muscles were active at the beginning of stance during overground locomotion and very low activity and kind of sporadic activity during the beam-like tunnel posture. Not at all as predicted. Just to highlight that. And where these lumbosacral muscles are. So again, this is the posture that should have the greatest amount of epaxial and hypaxial response to counteracting the increased moments at the shoulder and the pelvic girdle. And yet we see this very low level activity of those same muscles. Those muscles are more active during overground locomotion. And it was posited before that this kind of arched posture was maintained passively by ligaments. And now what we think might be happening is, in fact, this posture might be the one that is maintained by some passive elements as yet undescribed. So indeed, perhaps it's actually this posture that is the adapted posture. This is kind of the natural posture for a ferret. It can maintain that position. And it's not advantageous in the sense that it's, efficient or mechanically better in any way. It's simply what they need to do to fit into a tunnel and eat their dinner. This posture, however, allows them to efficiently traverse over land, in fact, so efficiently that they exceed what is predicted based on their mass alone. In conclusion, there are some emergent properties in biological systems that seemingly don't match our predictions. They don't match what we model or expect to happen. But these particular gaps are really exciting. And this is where we can spend a lot of our time. If you're planning on starting a dissertation or writing a grant, this is the stuff that we should spend more time in. These either little gaps like in senile sarcopenia or broader gaps in other systems. These are what I think are the most exciting parts of biology. This is where modeling and biology, to me, represent an interesting way to find even more gaps in our knowledge. And I try to question dogma and challenge too tidy examples as much as I can, but like every other aging professor, I find myself a little bit too comfortably slipping into the habit of saying this is simply the way that it is. So I challenge all of you younger researchers not to do that. And I'd like to acknowledge past and current funding resources, as well as current members of the Horner Lab, all of whom are great and contribute to a product that is definitely the sum of its parts.
Great. Thanks so much, Angela. That was fantastic. Does anyone have any questions?
Do you mind if I stop sharing?
Yeah, go for it. Yeah, then we can see all the pretty faces. So I have a question maybe to kick it off. Regarding your last examples there, the idea that really curved back posture for overground locomotion in the ferrets is so much more ideal, but the idea that the back muscles are actually working fairly hard to make that happen, the idea being that all the limb muscles are working less hard to make, like, for the actual propulsion components? Is that what's going on?
We should probably step back and consider, so the limb muscles, I of course didn't measure the activity of, but we should predict based on their posture that the limb muscles are working a little bit more in the crouched posture simply because of the crouched posture, the increased moment arms, etc. Also to factor in that moment arm distance, even within the limb, is going to change because the ground reaction vector should be directed toward the center of mass, although there's a little bit more recent research that suggests even that might not be true. But I should point out that the sacrospinalis and the multifidus in small mammals are used to kind of extend the lumbar region of the back almost as a contributing segment of the hind limb, if that makes sense. So if you're just a little animal and you're using your legs to move around, that's cool. If you're galloping, you're actually going to add quite a bit of dorsiventral flexion to that. And so what we see in animals that are traveling very quickly and bounding in particular, and things like squirrels, it's pretty easy to think about. The multifidi and sacrospinalis muscles are activating quite a bit for those forms of locomotion. So in a sense, they're a way to extend the portion of the body that is contributing to propulsive forces. But it was theorized, posited, that perhaps in this particular posture, they were going to just simply be tonically active and help maintain that beam-like posture.
Do they do that bounding behavior?
They sure do.
Gotcha.
Yeah. And other mustelines actually don't have that arch shape so much. They'll tend to just kind of noodle around when they're running around. But if you look at videos of different weasel species, they don't really tend to have that arched back posture. So it turns out that's actually kind of a singularity feature of the ferret and the Siberian polecat from which it's derived.
Gotcha. Great, thanks.
We have a question.
I think, we have a hand from Nick.
We've talked about this before, but I really like the passive force optimum length relationship. Do you have any thoughts on why, like, where passive force starts to rise and optimum length might be the same? Do you have any kind of mechanistic reasons why that might be true?
Mechanistic reasons for why passive force is sort of dictating operating length? Yeah, that is a really good question. I myself have not thought of any mechanisms beyond, the very sort of obvious. As soon as we start to see, say, 10% of P naught in passive tension, that perhaps the components that are responsible for, managing operating length will start to reduce. There might be some signaling that's involved as kind of a protective mechanism. Like, one of the interesting things to me is that we can tell a human, to maximally produce force, and they will do their best, and there's absolutely nothing they can do except produce that force. Then, of course, if we stimulate the muscle, we can get still more force. So there's a lot of kind of central nervous system drive that is missing in this, my particular take on it, and is certainly probably a big consideration of this. But there are these protective mechanisms that are not possible to detect if you're only looking at the muscle tissue itself, right? So we have these neural drive protective mechanisms that are not allowing us to produce that maximal force because it would actually hurt us. It would break bones, maybe. And that is the current state of senile sarcopenia, and finding the mechanisms that are contributing to muscle weakness seem to be mostly focusing on neuromotor considerations, as you well know.
Yeah, I was going to ask if you thought there was a sensory component, because I can see the kind of being like, you don't, like, a really slack muscle length is going to be hard to generate force because you're just doing internal work on stuff, and a really long length with high passive force is probably dangerous, but there's probably a range in the middle that's kind of fine, but you have like an integrated sensory motor system. So,
but yeah, that's an interesting thing to study, because we can't really parse that if we're only studying the tissues, right? So we do have to always have some comparison to what's happening in vivo, um, which is why it's always good to have a me and a Natalie on any given research project.
Because I refuse to do in vivo work. Thanks.
I think Nick had a question.
Nick, yeah.
Yeah, thanks. Thanks for a fun talk too. Um, uh, so I'm trying to bridge your first and your second aim, and
There's no bridge.
well, I want to make one though. Can we make one, please? So I don't know that much about multifidus geometry, but it strikes me that to go from like that much super curved to that much super flat has to be a pretty big angle change between the vertebrae, and presumably also a pretty big change in muscle length. So part A of the question is, do you think there is, knowing more geometry than that you do you think there is an appreciable change in length? And if so, is that going to be wider than, say, the plateau of the LT curve? Like, you're going to have to deal with
Hmm.
effects of significant length change?
There certainly should be a length change. So
But by
multifidus,
definition, right? Like,
yeah. So multifidus tends to span 2 to 3 vertebrae, sacrospinalis sort of similar, and they're restricted to that lumbar region of the vertebra. It's, there are not very many fishy epaxial muscles left in a mammal body, but these are actually not too far off because they do, simply go from bony origin to bony insertion, and again, only span a few vertebrae. So yeah, there should be significant length change. The fact that they are barely activated during tunnel locomotion, uh, yeah,
I mean, presumably at that point they're lengthened, right? Or are they? What side of the vertebrae
they,
are
they should be lengthened, I would think.
they? And so then, I mean, it makes sense that you wouldn't want to have them super active at a super long length for fear of damaging muscle. But um, I guess my overall question is this lengthening 3% length change, or do you think it's significantly more?
So I can't really comment on the percent length change, but that's actually that would be pretty easy to just do with a cadaver specimen, actually, just to measure the actual whole muscle system. It's a fairly simple muscle. It doesn't have a lot of tendinous inscriptions or any other annoying things that limb muscles do. So that would actually be a pretty straightforward thing.
Yeah, and you
Before,
could use the geometry of the bone to you could X from it and then just track attachment sites.
no extra for
Why not?
because I only have so much time. Yeah, the
But I
brown
mean, but
people
you could do it
always
with a
X
dead
around.
specimen, though, right?
Yeah.
Like,
No, and before COVID hit, Chris Law and I had actually talked about doing this a little bit more. I've actually, all my ferret work has kind of come to a screeching stop since coming to California because they're really hard to get permission to work on in California. But it can be done.
dead ones, work
Dead
on dead
ones,
ones.
yeah, dead
California's
ones are
not mad
pretty
about that.
easy.
Thanks.
Well, that's kind of also begs the question of an interaction between like doing experiments that bridge that gap, right? Is what about aging in ferrets and what are their muscles doing when they're, and how much of a performance decrease would you see the second they need to be operating at these longer lengths, at least in their back,
It,
being tennis?
there used to be a huge push in NIH for finding different models of aging because, mice and rats have been used over and over and over and don't necessarily actually model human conditions very well. But uh, it's, in terms of time, it's just very difficult because ferrets can live 8 to 12 years, 8 to 10 years, more like. So it's interesting because like other small mammals, including our dear pets, there tends to be a really steep decline in locomotor performance where your animal is doing fine from like all the way through 10 or 11 years old, and then suddenly they crash.
Mm.
And human decline tends to be a little bit longer. Um, but yeah, it would be really interesting. see if I can get ferrets for a long time.
I had another sort of follow-up question actually on Natalie's question, which was, have people tried to experimentally induce sort of rapid aging, like, or the effects of rapid aging, in the sense that, I mean, there is constant sensory feedback in order to prevent us from hyperextending joints or pulling muscles or damaging muscles, right? And as we age and as we're experiencing all of these various physiological changes that are impacting our muscle stiffness and everything, we have an inordinate amount of time to adapt how we move in order to prevent damage and move well. But what if there was a way to, I mean, I don't even know if there is a way, but what if there was a way to induce some of those effects acutely and see the effect before they've had a chance to kick in and adjust their locomotion to compensate, to sort of get at those kinds of, you could
I
tease
think
apart some things that way. I don't know if there's.
that's beyond the scope of what I know. And again, most of the aging literature tends to be in the biomedical realm. So it's actually hard, very hard to find aging study from like a more comparative biological perspective. But Stan Lindstedt has done quite a bit and has a book on aging that's pretty interesting. But the humble North American opossum is frequently used as a model of aging because it ages really rapidly, like 2 years, and it's donezo. And it's unexpected because you expect a sort of larger mammal to live longer, but they do not, and they tend to not have that aging period. Like they're fine, and then they're abruptly not. So if you imagine like our aging kind of begins, physiological aging for us begins, 35 or so, and then it's just this long, slow progression to death. Kind of dark here. But that, long, slow progression to death can be kind of fun and takes a lot of time, and we can still do a lot in the meantime. And in opossums, it's just like this very steep, sharp, very short time relative to their size.
Yeah.
Yeah, I mean, I think that's actually a tremendously big gap in biology, and why, I do tend lately anyway to spend a lot of time in biomedical literature. But most of the biomedical research is going to be geared toward bettering human health, building better human models, figuring out better strategies for humans to not age so rapidly. And we're kind of missing the idea of like, why do we age? There's certainly some genetic gene signaling, I think, that's been targeted in some studies, but that's beyond my expertise.
Awesome. Travis, you have a question. Nice to
Yeah,
see
so
you.
I don't, what's that?
Nice to see you.
Yeah, how you doing? So I don't study mammals, and so I have like some basic, super basic questions. When a ferret's in the tunnel, is their back pushing up against the roof, or are they holding it? Are they maintaining
That's a
that
good,
posture, or
yeah.
is the environment pushing that posture onto them?
They can maintain that posture. So we can, as soon as they enter the tunnel, they kind of adopt that posture before their body even enters it. And their body actually tends to be kind of slung a little bit low between those two girdles. But yeah, they can maintain that posture and sometimes will just behaviorally for no apparent reason. Kind of like if you have a cat and you try to put a halter on a cat or anything on its body, and they tend to do like that commando posture.
And so then are they flexing to hold that lower posture, or are they just not flexing the back muscles to hold them into that U shape?
That's the real question, and it turns out that our EMG study probably didn't have enough targets to really get at that. So we certainly didn't do any of the more cranial epaxial muscles. We didn't target any hypaxial muscles. Those are notoriously difficult to get. But there are certainly missing elements. So all we can effectively say with that limited EMG study is we thought it was this and it's not. And
And
that's
then to
hard to
follow that
push.
up, people, you were talking about using cadavers, right, for frozen or freshly euthanized specimens, when there's if you're looking at a fresh dead animal, what's their natural? Will a dead ferret sit in that U shape, or will they sit flat, or do they have
They're very,
no preference?
yeah, I mean, they're very floppy. They're absolutely, it doesn't seem
Interesting.
like there is a very stiff nuchal ligament, for instance, that's maintaining either posture, but that's what's posited. However, when they're in this arched position, as Nick was alluding to earlier, so that's going to create a whole bunch of different angles along the vertebral column. It might be that once we're in a position like that, it might be locked in. There was a researcher named Virginia Naples, actually, I don't know if she's retired or not, but she had explored some ligament that she found me at a conference many years ago and said, I have the thing you're looking for, but I still haven't seen that, so I'm not sure what happened to it. But she swore there was some ligament that was helping to maintain that posture. And I
Cool.
haven't
Okay.
seen anything obvious, but
Mhm.
it definitely needs more study.
Great. Well, maybe we should let people go. I know we've had a few people drop out because the hour has come. But if there aren't any more questions, thank you so much for a fantastic talk. That was really a lot of fun and a nice preview 1 month before, just over 1 month before sick bay when we get to see each other in person. So
Oh
really
yeah,
great.
sick bay is going to be boring biomedical tendon stuff.
Oh,
This is more fun for me to think about.
yeah.
Yeah, this is actually a really great excuse, Kathleen, for me to pick up problems that I put down a while ago, especially the ferret thing. Like when we didn't find what we wanted to find, I was like, oh shoot, what am I going to do? So this is exactly what I wanted to get out of it. So selfishly, thank you.
Awesome. No, thank you very much. And Alessandro put the website link in the chat, but it was also on the tweets and stuff. So anyone who's interested. But yeah, no, thanks a lot. And we're gonna have a bunch more next semester as well, too, to keep it going. So
Yay.
yeah.
Good
Thanks a
to see
lot.
you all.
Thanks a lot. Good to see everyone, and I'll stop the recording.